Welcome back to another episode of Facilitate TV. I'm joined by Dave, the CEO of Nucleus Biologics. Thanks for being here.
Yeah, thanks, Ben. Appreciate it.
Excellent. So just before we started the tape rolling, you mentioned that you guys are basically developing Nespresso for media cell culture.
That's right. That's exactly right. So
Tell me more.
Yeah, so we, first of all, I want to say thank you to the Advanced Therapies team. It's been a great conference. Peter Harkness, the rest of the group have done an amazing job. We launched the product, the Krakatoa, here yesterday in the innovation zone, and the system is very unique in that it's the first pod-based system for media manufacturing, and it allows you to make anywhere from 35 liters to 500 liters at your point of use.
Well done.
Yeah, thanks.
Okay. Yeah, so that's one heck of a big Nespresso machine.
Yeah. Yeah, you'll be on a caffeine high for a long time.
I guess so.
Yeah.
Excellent. So right, like, and this is such a big need, right? Because what does the current supply chain look like for getting media into your clean room?
Yeah, so I think it's one of the challenges that we struggle with in the entire industry is the whole model was built on traditional classical media manufacturing, which was 10,000 liter scale. You make it in bottles, you ship it out. It's all cold supply chain. You have to store it in refrigerators. Right. You have to have validation, especially for GMP, of every step in the process. So it's an extremely capital intensive, extremely greenhouse gas intensive process. And really what we have to do is rethink that entire process and disrupt it with something new, a new model.
Gotcha. Yeah, so what does that look like now?
So the name of the product is Krakatoa because we named it after the volcanic eruption of 1883, which is the loudest sound heard on Earth.
Nice.
And we feel like it's that disruptive. The concept is you have a formula, you let us know what that is. We will put that formula into a pod, send that pod to you, you pop it into the machine, and then you hook that machine up to any kind of ultra pure or Wi-Fi water, and you can make anywhere from 35 liters to 500 liters of media in under 3 hours.
And that can go right inside of your clean room, correct?
Yeah, you can do it in your clean room. It's a closed system, so you don't have to do it in your clean room. You can also do it in, an uncontrolled space.
Very interesting. Yeah, because literally yesterday I was talking with a cell therapy developer, and she was saying one of the biggest contributors to the cost of the therapy is the cost of the media. And so what's the cost savings we're looking like with something like this where you don't have the cold supply chain and whatnot?
So you're probably the way to think about this is it's probably 30 to 50% reduction in cost. And what you're eliminating is not just the cost, but you're also eliminating all the time to manufacture the media, wait for it to release through all the USP testing, and then ship the media. So that process could be weeks, if not months, and you're able to do this in a day, basically.
Phenomenal. Right, and so what are some results that you've seen so far?
So the response here has been fantastic. We already have multiple companies that have expressed interest in purchasing a system. We had a small scale version of this system that's a lab bench scale that only makes half a liter. That was our proof of concept, and the response has been great. I think people in general are looking for solutions to the problem of cost, and for us, it's about our mission statement, which is speeding the time from discovery to cure. And in order to do that, we have to include cost in that.
Yeah.
If we want to increase patient access to these life-saving therapies, we've got to address this cost. And as you said, media is anywhere to from 30 to 40% of the COGS of any cell or gene therapy.
Makes sense. And I'd be curious as well, right? Like, how do you incorporate this into the rest of the process with regards to like digitalization of process and everything related to GMP documentation, that sort of stuff?
Yeah, great question. And so we actually have a digital batch record that we generate from our machine. So, and then ultimately where we're going with this and kind of as we look out in the future, we want to be able to have a feedback loop with the bioreactor. So if you know that you're getting certain readings in your bioreactor, then you can change the components in your pod and feed exactly what the cells need at that particular time. Right. That's ultimately the idea, is that you create a closed loop feedback where you're modifying your media because it has such a profound effect on the cells.
Definitely something that the industry needs. We need some disruption with regards to really meaningfully reducing these costs. So congratulations, and I look forward to seeing where this goes.
Yeah, thank you. Appreciate it. Yeah.